Hello everyone! This module is brought to you by the MT/MV instructors of Angeles University Foundations. My name is Romania S. Salazar, your module writer for Module 4, An Introduction to Virology. At the end of this module, the student should be able to define the basic and common terms and expressions in relation to the study of viruses. We should be able to identify the general and distinguishing characteristics and structure of a virus. Also, we should be able to understand the general principles of viral replication in relation to its cycle. And finally, understand the general principles in the development of viral infections. To attain these objectives, our module topic includes an introduction, viral nature, viral replication and genetics, and viral pathogenesis. Virology is considered to be a subfield of microbiology or medicine, which of course is the study of viruses. Virus is an infective agent that is typically consists of nucleic acid molecule in a protein coat that is too small to be seen by the light microscope, and which is able to multiply only within the living cells, or the host. This slide represents an outline of the history of the most deadliest pandemic, and as we can see, from smallpox to Spanish flu, HIV, Hong Kong flu, yellow fever, MERS-CoV, Ebola, and up to the COVID-19, which have changed most of our lives. These are all viruses. And looking at these timelines, they have been with us through hundreds of years, and as the human race progressed, so does these viruses. Additionally, we can see that this virus has the power to wipe a chunk of our population. According to a study in August 2020, here are the most deadliest viruses over the last 100 years. And we can see through these numbers that we should not take this virus for granted because it can really infect and kill human race. Take a look at dengue virus from 1950 has already infected 100 million to 400 million people in a year and has a 2.5 mortality rate. Look at HIV. From 1920 has already infected 75 million people, and out of these 75 million, 32 million have died. We can conclude that we are continuously learning about the unpredictable power of nature. This is nowhere more true than in the continuous evolution of new infectious threats to human health that have emerged, often without warning, from the natural environment, and some also re-emerge. And even in the 21st century, this will continue to emerge and re-emerge. So we have to take note of what reproduction number is in this slide. So yung reproduction number is a measure of how many people will a sick person can infect in an average. So let's take for example the transmission of SARS from China to Hong Kong in 2003, which took several countries affected. So what happened is it started with a person from China infected with SARS, went to Hong Kong, checked into the hotel, and into the hotel exposed other people on it. So ang nangyari is that when he was still in the hotel, felt sick already, went to the hospital, and into the hospital right away, he exposed to him 4 health workers, to whom these 4 health workers got in contact with other 56 people, including health workers and patients. And again, these people got in contact with other health worker in 4 different hospitals in Hong Kong. So if you will trace back, siya palang mag-isa, including the family members, ang dami na niyang nahawaan. And then, let's take another example. So, itong si patient B. So, si patient B, again, got exposed to him, went to the hospital. Maybe he's asymptomatic kasi, and he's not infective yet kasi hindi siya nakahawa ng health worker in it. Maybe he got cleared, that's why he went home to Vietnam. However, when he got into Vietnam, maybe he got hospitalized and got this 37 health worker exposed to him or infected with him. And another health worker infected another one who went to Bangkok. So, nakarating na ng Bangkok. So, from China to Hong Kong to Vietnam to Bangkok, nakarating na yung virus. Let's see another example. So, let's say itong patient CDE. All of them are Singaporeans, no? So, they didn't went to any of these hospitals. So, basically, they went back to Singapore, and when they were in Singapore, they went maybe to the hospital kasi meron silang na-infect na 34 health worker. And this 34 health worker got close contact with other 37 people. And one of the health worker who got exposed to them was exposed to another health worker who went to Germany and of course infected their families also. So same happened to a person from United States, a person from Ireland, and another 2 persons from Canada. So ang bilis kumalat, 'di ba? Especially if the virus is so infectious, there will be a continuous spread of the infection if we will not be cautious of the common knowledge with these viruses. Kaya nga, di ba, during the COVID-19 pandemic and even right now, because we don't know anything about COVID, syempre at first, no, the solution was to have social distancing, di ba? So that to prevent the spread of the disease. So what is a virus? A virus is a small collection of genetic code, either a DNA or RNA, surrounded by a protein coat. They lack both cellular structure and they are independent when it comes to metabolic processes. A virus cannot replicate alone. Viruses must infect cells and use components of the host cell to make copies of themselves. They replicate solely by exploiting the living cell based on the information in the viral genome. Often, they kill the host cell in the process and cause damage to the host organism. They are autonomous infectious particles that differ widely from another microorganism in a number of characteristics that will be taken in consideration in the following slides. So, the general characteristics of these viruses are, they are non-cellular, meaning they do not have properties of a cell or organelles that can support their lives. They are obligate intracellular parasites, which means they resemble attributes of a parasite since they need living cells in order for them to live or reproduce. Thus, the term parasite is taken up. The size range from 20 nanometer, which is the poliovirus, to 300 nanometer, which is the pox virus. That, although they are very small pa rin, they cannot be seen even with a light microscope. So, to gauge their size, they are relatively smaller than the average size of a bacteria, organelles, and eukaryotic cells that are normally seen in a light microscope. And that's why earlier I have mentioned that these viruses are relatively small, that they are only seen using an electron microscope. So their composition is made up of nucleic genome, either be DNA or RNA, and also with a protein coat which is called capsid. Since they do not have cellular properties and A nucleic genome and a protein code serves as their basic composition. So, the nucleic genome consists of only one nucleic acid that can either be RNA only or DNA only, depending on the species. And for the capsid, it is a protein coat. Furthermore, because they do not have a cellular composition, nor do they grow or metabolize other organic matter or materials, they produce no waste products. They utilize no energy, and they do not adapt to their environment. And in addition, they reproduce, but only within a living cell. That's why they are said to be not living thing, but have properties of a living thing. So how are viruses being classified? There are a lot of characteristics that makes virus different from others. But for the purpose of this module and the succeeding discussion, we will differentiate these viruses according to this basic characteristic only. So first is your genome. Whether what type of nucleic acid is present, either DNA or RNA genome, this is very important and a basic differentiation of the viral type, as well as it is a configuration of which of the nucleic acid structure is present. So, whether it is a single-stranded or double-stranded, ma-identify natin what type of virus it is. For the RNA naman, in addition, there are further subclassified according to plus and minus polarity pa. Next is your capsid symmetry. This refers to the shape of the virus. So, ang mga shape natin would be cubic, helical, or complex symmetry. Another is the presence or absence of an envelope. So, an envelope is made up of lipid. So, if the lipid is present, ang tawag sa kanya is envelope. If it's absent, it is non-envelope or naked. And also, in addition, we will be able to differentiate them according to size. So, diameter of the viron or of the nucleocapsid with helical symmetry will give you a hint of what type of virus is present or what type of virus you are looking at. So, what are capsid? So, as mentioned earlier, capsid is the protein shell or coat that encloses the nucleic acid genome. Basically, they are for protection of the nucleic acid. And in addition, they assist in the attachment of the naked viruses or the non-envelope. Also, in some viruses, your capsid serves as an enzyme. Next is your nucleocapsid. What is nucleocapsid? This is a protein nucleic acid complex representing the package form of the viral genome. So, it is a capsid plus a nucleic acid genome. Another is your capsomer. These are clusters of polypeptides seen in virus with icosahedral symmetry, which is the morphological units that can only be seen in the electron microscope. This should be on the surface of a virus. Though they represent clusters of polypeptides, the morphological unit do not necessarily correspond to the chemical definition of the structural unit. Next is your envelope, a lipid-containing membrane that surrounds some viruses. Kaya nga yung iba, envelope, yung iba, non-envelope. It is acquired during viral maturation by a budding process through a cellular membrane. Virus-encoded glycoprotein are exposed on the surface of the envelope, and these projections are called peplomers. This envelope are also responsible for attachment, entry, and assembly of matrix protein. And lastly, A viron is a complete viral particle. It serves to transfer viral nucleic acid from one cell to another, and in some instances, example are the papillomavirus and picornaviruses, the viron is identical with the nucleocapsid. And in more complex virus or virons, like herpes virus or orthomyxoviruses, this includes the nucleocapsid plus a surrounding envelope. And the structure of the viron serves to transfer the viral nucleic acid from one cell to another. This slide illustrates the viral structure. This one is an envelope virus with icosahedral symmetry and a virus with helical symmetry. So to understand the viral structure, look at this picture, class. As mentioned before, viruses do not have organelles. Therefore, merely they depend on the host cells, but the basic structure of the virus is a nucleic acid genome. The nucleic acid genome can either be DNA or RNA, which is enclosed by a protein coat called capsid. And these two, the nucleic acid genome and a capsid, is collectively known to be the nucleocapsid. Although not all viruses have capsomere, some have this capsomere or clusters of polypeptide around the nucleocapsid of the virus with icosahedral symmetry. Again, not all, but some of the virus may have lipid structure, ito yon, which is called the envelope. Kaya nga yung iba, non-envelope or naked, pag wala silang lipid structure. Pero pag meron sila, they are called envelope. Okay? So, in addition, some viral structure can be seen with glycoprotein spikes, which aids in the attachment of the virus to the host receptor. So, all in all of this together make up a viron, or a complete viral particle that can exhibit reaction in the host cell. Now, if you will look at this slide, you will see the viral morphology or the viral architecture in viruses. And it comes in three different morphology. You have the helical, icosahedral, and a complex. So your helical resembles a helix or spiral or spring, while your icosahedral, we can see a 20 faces of equilateral triangle. Ito siya. And in complex, it is described to have a more complicated structure or shape. So, pwede siyang box shape or any other shape besides the helical and the icosahedral. So, these are the example of viruses with this morphology. So again, these are the shapes of the viruses. So these are the DNA viruses. These are the RNA viruses. Example of icosahedral are the herpes viridae. Example of helical are the one that causes rabies and the tobacco mosaic virus. And for the box shape, example is the pox viruses. Now, for the viral composition, we have different composition of the viruses. Meron tayong protein, nucleic acid, lipids, and carbohydrates. For the viral protein, this facilitates transfer of viral nucleic acid from one host to another. It also serves to protect viral genome against inactivation by nucleases. It participates in the attachment to a susceptible cell. And lastly, provides structural symmetry of the viral particle. For the viral nucleic acid, as mentioned earlier, it could either be DNA or RNA, which encodes the genetic information necessary for replication. And for the lipids, the one responsible for the viral infectivity, that is why when this is dissolved, infectivity also disappears. And lastly, for the viral carbohydrates, it appears to be in the viral envelope in the form of glycoprotein that is responsible to the attachment of viral particles to the target cells in the host. But nevertheless, not all of the virus has these glycoproteins. Let us take a look at this SARS-CoV-2 structural component for us to be able to compare the previous discussion for viral proteins. So, your membrane protein, which is represented by letter M, is the most abundant of them all, defines the shape of the viral envelope. The central assembly of the structural proteins also is due to the membrane protein. Another is the envelope protein, which is represented by the letter E. So this is the smallest protein among them, and this is responsible for protecting the viral component. And then you have the S protein for the spike or the corona or the crown, which is for attachment to the host cell. You also have your nucleoprotein, which is responsible for transferring the nucleic acid genome to the host cell that could hijack the host cell for them to replicate, resulting to increase viral load and increase the probability of infection. And you also have your nucleocapsid with nucleic acid, and in this case, this is an RNA nucleic acid in the capsid, ayun sa loob. So why is it important for us to know this, class? It is important because this structure and composition is what our scientists are looking at in the formulation of vaccines, and also to know which among these parts serves as the weakest link or the weakest point of these viruses, so that we can easily develop an antidote which attacks this weakest point. Because once this weakest point is further attacked, there will be a disruption of the protein and thus the envelope, and thus resulting to death of the cell. Diba? So let's take for example yung hand washing with water and soap. This disrupts specifically the E protein or the envelope protein. Thus, when the envelope of the virus is disrupted, it will shorten the lifespan, decreasing the potential of this virus to be more infective. Viruses multiply only in living cells because they need to utilize the energy and take over the host cell's machinery to be able to synthesize new viral particles. So there are different methods employed by the viruses to be able to make more copies of themselves. Though the specific details of these methods may vary from one method to another, there are general outline for the replication cycle of the viral replication. So meron tayong specific steps for this. They are attachment, penetration, uncoating, replication, assembly, and release. So, during attachment, the viral proteins on the capsid or phospholipid envelope interacts with the specific receptors on the host cell surface, or endocytosis happens. So, the specificity determines the host range or tropism of the virus. And then eventually, the second step is the process of attachment to a specific receptor that can induce conformational changes in the viral capsid protein or in the lipid envelope that results in fusion of viral and cellular membranes. So some DNA virus can also enter the host cell through receptor-mediated endocytosis. And then third step is the uncoating. So during uncoating, the viral capsid is removed and degraded by the viral enzymes or the host enzymes, releasing the viral genomic nucleic acid. And once the nucleic acid is released, replication happens. So, transcription and translation of the viral genome is now initiated. It is in this stage of viral replication that differs greatly between DNA and RNA viruses. So, basically, after that, they will replicate and replicate themselves. So, after they have replicated, assembly will happen. So, assembly meaning the synthesized or the replicated viral genome and proteins will now be post-transcriptionally modified. So, viral proteins are packed with newly replicated viral genome into a new viron that are ready for release from the host cell. So this process can also be a time for them to mature before they are released. So finally, when they mature, release or the sixth stage will happen. So for the viron release, there are two methods. So release by lysis or release by budding. So when you say lysis, this results in the cell death kasi what will happen is it will burst. Okay? But for the budding, what will happen is it will just bud out. Okay? So those are the different viral replication steps. There are two modes of viral replication or viral survival strategies or the methods. Meron tayong lytic and lysogenic cycle. So for the lytic cycle of the viron, it invades the host cell, ultimately resulting to killing them by bursting the cell while spewing the virus out of the cell ready to infect other hosts. So as the name connotes, lytic, meaning the cycle will always end in the lysis or bursting of the host cell after all the steps in the cycle has finished. So remember in the previous discussion, we said that the viral replication somehow have the same steps. So as the lytic cycle follows the step previously discussed. So yung first step natin, attachment, second is penetration, third is biosynthesis wherein the virus starts to utilize the host cell's machinery, replicating itself, and the fourth, as the number of your virons increases and matures, the newly synthesized viron assemble itself for release. So as their number increase, the host cell weakens and eventually step 5 will burst or lyse the cell, resulting to death, releasing the virons produced ready to infect other cells or hosts. On the other hand, in the lysogenic cycle, a viron's DNA or RNA is integrated in the host cell genome, thereby becoming a part of the cell. Meanwhile, the viral particles will replicate together with the host cell genome, undergoing viral latency or a period of dormancy, where it just replicates in the host and stay there without even harming the host cell until a stressor or activating factor arrives. Again, what will happen is your lysogenic cycle will get back to the lytic cycle in an overlap step. So yung mga steps natin is almost similar with the other. It starts with the attachment and then penetration and integration of nucleic acid genome with the host cell, which becomes a part of the host cell genome, now integrating a fragment of its nucleic genome to the host cell. So pag nag-replicate siya on the third step, it will get along with the host cell, divides for as long as it can, and remains there without hurting the host cell. And then the fourth step, which is the latency or dormancy, until such time that the triggering factor will prompt a reactivation, nandun lang siya sa latent stage. Okay? And then eventually, it will just replicate there since the viral fragments is incorporated in the host cell genome. When the host cell divides, the fragment of the viral particles in it also replicates and continuously until such time the reactivation arrives. So for example, ng mga stressor natin or reactivating factors or chemicals, carcinogens, UV light, low immune response, yung mga yan. So, the viral particles in the host cell will now detach kapag the stressor arrives. So, it will detach itself from the host cells. So, yun yung fifth stage natin ngayon. That is now undergoing lytic cycle of replicating itself until a new viral particles are produced enough to assemble itself, getting ready to burst the cell, causing the burst or release of this replicated virus into the environment, ready to infect other cells. Next is the viral pathogenesis or the viral infection. This is the process that occurs when a virus infects a cell and causes changes in the cellular characteristics. Take note that in any disease, infection, or pathology, there will always be three main factors that will be involved. We call it an epidemiological triad. So we have the host, the environment, and the agent. So the host, including us, may include but not limited to the host age, gender, genotype, nutritional status, health status, and even behavior. For the environment, it includes the weather, housing, geographical location, occupational setting, air, and even quality and availability of food. And lastly, the agent factor, which includes its infectivity, pathogenicity, virulence, immunogenicity, antigenic stability, and of course, survival. And the success of a disease or pathogen, whether it is caused by whichever of this agent, could be a virus, parasite, or a bacteria, is an interplay of these three factors. So, hindi po pwedeng isa lang. So, dapat these three are present in order for you to develop a viral pathogenesis or a viral infection in this case. This slide shows you the general principles on how virus causes infection in a host. So first is that many viral infections are subclinical, meaning not all viral disease caused by viruses are severe. More often, they are self-limiting or asymptomatic, diba? Which means you can overcome it by your body defenses only and mechanism. So for example, when you had a viral flu, you'll be visiting your physician. And your physician will not right away prescribe antiviral or antibiotics. The first thing your physician will tell you is that, oh, you take a rest and a lot of fluids muna. And after 3 days, if you don't improve, that's the time you take this or take that. It is because of the same reason that these viral infections are self-limiting. Next is that same disease syndrome may be produced by a variety of viruses. So, ibig sabihin, if you look at the different signs and symptoms of most of the viral infection, you cannot single out a specific sign or symptom specific for a certain virus. So, their symptoms could have similar and generalized manifestation, parang ganon. So, for example, no, fever, chills, body fatigue. This could be a symptoms of whether you have a coronavirus or an influenza virus. Third is that the same virus may produce a variety of disease. So, a perfect example is the coronavirus. though they are under the same family, they could cause different types of diseases. Example, no, yung MERS-CoV, which is the Middle Eastern Respiratory Corona Syndrome, SARS-CoV, Severe Acute Respiratory Syndrome, at ayan na nga, yung COVID-19. All of them are under one family but produce different diseases. And fourth is that the outcome may be multifaceted. So, as mentioned earlier, that meron kang host, environment, and virus. Diba? And the outcome of the infection in any particular case can be determined, but the interplay of these three factors are actually important and interrelated with each other. So, hindi po pwedeng magkaroon ka ng sakit without these three factors. So, similar to the viral replication cycle, there are also general steps in the viral pathogenesis, no? Always remember that viral pathogenesis are the different processes that takes place from the time that the virus attaches itself to the host receptor cells up to the time that the host exhibits signs and symptoms of the disease, then eventually up to the recovery or sometimes to the death of the cell itself. So just like the viral replication, there are different steps. So these steps are, first, the virus attach and enter the cells of the body surface and then replicates to produce new virions ready to be released. And then the second, the mechanism of viral spread vary, but the most common route is via bloodstream or the lymphocytes or the lymphatics pala. So viruses have different mechanism in spreading infection. Some may produce infection right away in the port of entry, while others may spread systematically to bloodstreams or lymphatics, which is called viremia, or the presence of virus in the blood. And then third is the destruction of virus-infected cells in the target tissues and physiologic alterations, which are responsible for the development of the disease. So during this, cell injury is actually happening, wherein the hosts develop the disease through signs and symptoms also are manifested during this stage. And the fourth one is when the host either succumbs or recovers from the viral infection. The immune system of the person has a great influence on this. So following the infection, remember that the host may either succumb or fully recover through immune responses or adaptive mechanism. But take note that this may vary from one individual to another, or because of the three factors mentioned earlier, yung host, agent, and environment. And lastly, the last stage of pathogenesis is when infectious virus is shed into the environment. Shedding usually occurs from the body surface involving viral entry. So this is necessary to maintain a viral infection as the infectious agent expels and released to the environment after a successful replication to a new host naman. Therefore, it could be understood that viral shedding and infective stage are synonymous to each other, no? Though this stage could be different from one virus to another. So let's take, for example, the pathogenesis of poliovirus as an example natin, ano? In order for us to understand better these steps, recall that your poliovirus is acquired via oral fecal route. So it begins in the ingestion of the virus from contaminated water or food. So then, the virus invades the regional lymph nodes and develop viremia. Or the virus spread to the different parts of the body through the bloodstream. If the virus invades and overcome the immune system, the virus will now replicate in the horn cell, specifically in the horn cell, causing cell destruction, motor neuron damages, and even paralysis. And then, the individual infected with the poliovirus will shed off some of the virus through the excreta. That is why there's a need of clean water and sanitation, diba? This is one of the sustainable goal of the WHO, specifically number 5. It is one of, or the primary aim in the community development, which is to eradicate open defecation or prevent contamination of sewerage and water so that polio, the infectious agent, could also be eradicated. So as described earlier, infectious diseases result from interaction of the agent, host, and the environment. But more specifically, transmission occurs when the agent leaves its reservoir or the host through a portal of exit, then is conveyed by some mode of transmission. Mode of transmission or enters through an appropriate portal of entry to infect another susceptible host. This sequence is sometimes called the chain of infection. So for the mode of transmission, this is a very important aspect in understanding viral pathogenesis because this actually can help us design an effective mitigating measures against the virus. So, pag alam mo kasing it could be transmitted to direct contact, edi prevent direct contact if you know that the transmission is in droplets. So, you have to wear mask. Mga ganun. So, there are different modes of transmission of the virus from the reservoir to the susceptible host. So, according to your CDC, it is classified into two: direct transmission at saka indirect transmission. So, for the direct transmission, an infectious agent is transferred from a reservoir to a susceptible host by direct contact or droplets. So, pag sinabing direct contact, this occurs through skin-to-skin contact, kissing, sexual intercourse, mga ganon. And direct contact also refers to contact with soil or vegetation which harbors infectious organism. So, example natin dyan, yung infectious mononucleosis or the kissing disease. Sa mga bacteria naman, yung gonorrhea spread through person to person by direct contact. And also, sa parasite, example natin is the hookworm, which is spread by direct contact also with contaminated soil. For the droplet spread, it refers to the spray with relatively large, short-range aerosol produced by either sneezing, coughing, or just talking. So the droplet spread is classified also as because transmission is by direct spray over a few feet before the droplet falls to the ground. So example natin dyan ayan na nga, yung COVID-19. So another example, yung pertussis. Another is meningococcal. So these are example of diseases transmitted from infectious patient to susceptible host by droplet spread. So in indirect transmission naman, this refers to the transfer of an infectious agent from a reservoir to a host by suspending it into the air particles or other inanimate objects or intermediaries, which are referred to as the vectors. So, pag sinabing airborne transmission, it occurs when infectious agents are carried by dust or droplet, which is suspended in the air. So, yun yung difference ng airborne sa droplet, no? Pag airborne, the particle is still suspended in the air. So, next is your vehicle-borne. So, when you say vehicle-borne, it may indirectly transmit an infectious agent, including your food, water, biological products, fomites, yung mga inanimate objects such as doorknobs, bedding, scalpels, yung mga ganyan. For the vectors, this includes yung mosquito, fleas, ticks. They may carry an infectious agent through purely mechanical means. They just have it in them. Pag naman sinabing biological vector, it means they support growth or changes in the agent or certain stage of the growth of this agent has to be in the vector. So, yun yung mga differences of mode of transmission. Lastly, we'll be discussing the viral effects on the host. So, first of it is the cytopathic effect. As the name connotes, cyto means cellular, so there is a change in the cellular morphology caused by the infecting virus that can be a change in size or shape. So, if you look at this picture, makikita natin itong three large cells. Most likely, they are infected with type 2 pneumocytes with a single prominent red staining nuclear inclusion surrounded by a clear halo. So, if makikita nyo, this one should be the same as this one. Pero, you will see there are prominent nuclear staining bodies or inclusion, tapos enlarge, and then meron pang halo dun sa gitna. So, the morphology is totally different. Okay? So, next is the transformation. So, in transformation, the cell undergone multiple changes that covers a normal cell into a malignant one. This often happens in cancer-causing viruses. So, includes here are the inhibition of cell, macromolecular biosynthesis, alteration of plasma membrane, and also your lysosomes. And lastly, development of inclusion body. So, in this second picture, makikita natin here, you will see an epithelial cell revealing a characteristic of polyomavirus infected cells containing an enlarged nucleus. So, siya yan. For the latent infection, this occurs when the infecting virus persists in the body and reactivates to an infectious virus and produce clinical diseases. A very good example is your herpes zoster or the shingles. So, kung makikita nyo dito, there, once you had the chickenpox, di ka na magkakaroon ulit ng chickenpox, di ba? Although, the virus is with you or just sleeping or on the latent stage in you, and then somehow, when some triggering factors affects or came to be affected by the triggering factors, this will reactivate and it will reactivate as a shingle. Okay? So, though some are asymptomatic, yan, at first, tapos pag bumaba yung immune system, sabi ko nga yung tatlong factors, nagsama-sama sila, again, it will exhibit another infection. And then lastly, the cytolytic effect or cytocidal, which means virus kills the host cell. So, cell death na ang mangyayari sa fourth, which is the cytolytic effect. Here are my references. And for your questions and queries, please feel free to contact me through any of this platform. Thank you very much for your time and happy learning.